Okay, so you should be back to home screen, DNA Subway. All we're going to play with today is the red line, okay? Annotate a genomic sequence. And so we're not going to do much more beyond that. And we're going to use, so you'll go here first, and you kind of come down, and you're going to use the Web Apollo, browser only, no Java. Note it does say better with Firefox in the bottom corner. I'm using it with Chrome. So far, it seems okay. So click the Web Apollo, and you don't need to follow these steps exactly right now, although you certainly are welcome to. We're going to do a plant, of course. And for the tutorial, we're going to follow their tutorial, and we're going to go along with the dicotyledon. Okay, so no big crops. We're going to go with the Arabidopsis. The very first one, Arabidopsis thaliana, mouse ear crest. That's chromosome 1. We have a 75 kilobase segment of DNA sequence we're going to play with or work with. So what you'll do is click on that. And then over here, you'll give a title. You can write trial 1, class 1, whatever you want to. Mouse ear, you can name it after you, name it whatever you like. But give it a name for yourself. But you know it's a testing. And it already imported the name over here, as you'll see. And you can give a description. Plant molecular biology, first trial, or I'm going to say first tutorial, whatever you want to. And continue. And when you push continue, then you'll have your choices, of course, and you'll go right on down. I guess I might as well do this. This is for me, probably like trial 3. Let's just try that. So you press continue, and you'll have all your tools here, okay? So the sequence is in there. What you just did when you picked the Arabidopsis organism, 1 chromosome, that was already, there's already a sequence for that. Normally at that point is where you would upload your sequence to work with, but we're not going to upload our own raw sequence. We're going with one that's already been here. So if you want to look at your sequence, you just click on that blue sequence at any time and they'll show you. That's all it is. Oh, it's a sequence. Look at all those T's. What do T's tell us? Like a lot of repetitive T's. Anyway, when you're done with that, you just close it if you just want to see it. You can also save that as a text file on your own computer. So we want to get rid of repeats. You'll click the repeat masker. It'll show you that it's running, flashing yellow, and when it's ready, you can view it, and you can click on it, and you'll be able to view. And the tutorial is going to take you, and it's going to tell you exactly step by step what to do. But this is what you can expect. You'll click that. And when you're done, you're going to try to predict genes. You're going to use software. You can do all 4 of these. For sure, try Augustus and I believe a tRNA scan. And so they use their own algorithms. Each software has its own algorithms for what it's looking for. Some are better or stronger than others. Just depends on your sequence and what you're looking for, so forth. But it'll try to predict some genes for you. And then you'll compare your gene to see if your gene is of any, has any homologs. And so we can get to the possible function of that sequence, the protein that it codes for. And so you can do a BLAST-in search for nucleotide or BLAST-X, which is probably what we'll do, and so forth. So the tutorial we're going to do will take you through that. So how do you get through the tutorial? Again, I'm going to just go back from home. So right back down here, if you haven't already, manual. So what you're going to have to do, you're going to have to log in or come back twice because unfortunately the manual does not open in its own window. So click manual. And there it is. This is a complete DNA Subway guide. So if you scroll down, you've basically already gone through this. You registered. If you want to see the difference between private and public, you can. So you're going to do a walkthrough of the DNA Subway red line genome annotation. You can either click there or just scroll down. It's the first thing. And so here's the DNA Subway red line. Create an annotation project with Apollo. Okay, and this is where it starts right there. Read every note, read every tip, go step by step. You'll see it goes pretty quick. If I go back 1, so you're going to go through all this right here. Create an annotation project with Apollo, find and mask repetitive DNA, making gene predictions, visualize predicted genes in a genome browser. All this right here, total will probably take you 15 minutes, maybe 20 tops. And then, and you can play around a little bit from time to time. Research database using BLAST. This could take a little bit of time if you're just playing around. Build gene models using Apollo will be the last thing you do. All these things, you don't have to keep coming back to that menu and clicking on it. You can see right here, DNA subway red line, visualize predicted genes in a genome browser, tells you what to do, what it's going to look like, some tips. Those sample exercise we're doing, results you can expect to see. Then you will copy and paste these genes when you are searching the database. So it gives you real step-by-step instructions what to do. So best is you'll have to kind of have that and then go back to DNA subway and go back into your, once you log in, if you've already started, you can go to My Projects if you'd saved it. And you should be able to click on your trial 3, and it'll take you right to where you left off. Okay, so now I can go back and forth between the instructions. And this is probably where you want to do a split screen. So pull that web page out. I don't know if you can still see that or not, maybe not. And just go, you know what I'm saying. Anyway, go half and half screen. I'd have to share 2 screens. It's not allowing me to share 2 screens. I guess I could share my whole screen. There we go. I think my whole screen is being shared now. So, so then you can have the, can you all see that? Is that showing you side by side? Anyway, that's pretty much it. Okay. So all the instructions are right here. It may be tedious because we've never used it, but it's good to have tedium because it's going to be literally step by step. Now, previously, I required that you answer these questions on paper, having a third doc open. I am not going to. Congratulations, you're welcome. But you ought to answer them at least on scratch paper or in your head. And what I'm referring to is when it asks you, like right here, when you've gone through the example exercise using the Arabidopsis chromosome 1, okay, it tells you what tool you're going to use, the repeat masker. Tells you the concepts you're covering, non-coding DNA, that's why you're repeating it, sequence repeats, mobile genetic elements like transposons. You want to get rid of all that because the whole point of this exercise is finding a gene to annotate, which means we're not interested in non-coding DNA, period, right? So, I mean, just glancing through each of these, but answering these questions for yourself or on paper. How many hits were detected in your sample? Look at how many hits there are. Average length of those. These will create real learning for you as you go through the steps and cause you to think about what you're doing so you're not just going through the motions, okay? If you do want a bird's eye view, go back to the, where was it? I think here. Or not. Anyway, and you can see exactly just what we're doing. Finding repeats, predicting genes, searching databases to find homologs, in order to get an idea of what the function of that sequence is that we have located or found, okay? Any questions? Hey, you got a glossary here. Okay, then. So there's your DNA subway guide. I believe I've said all I can or will for this section. And I'll leave you, it's 4:40, yeah, about 20 minutes. I think 20 minutes you can probably get through for sure half, maybe even 3/4 of it. So I apologize for that. You can do it to completion now. If you can say this, you just saw me open it and go right back to it. So if you do run out of time, or if you got to go, whatever you got started on, when you click back on it, your projects, My Projects, it'll pick up right where you left off, okay? These are some other things you've seen. I've done. Capiche? All right, I'm gonna pause for a bit. I'll stay on for questions. I got a couple thumbs up, and said I'll be here. I guess I'll be here. I'll be here for the next 20 minutes or so, or until I see everyone has left. I'm here. Okay, and in you run into stumbling blocks. Oh, I gotta stop this record. Actually, I am gonna have to end this meeting in order to, let me see.